I'm actually surprised there's such a large crowd because it's been so many months since the Ukrainian war conflict, and all of our attention just has shifted from COVID to what's going on overseas. I can say personally, all of my attention is there. I am Romanian. You'll notice from my last name, the ESCU at the end. We still have family there. They are housing refugees, and they're right there on the front line. But God is good, and the people that live there, they say they see miracles and God's provision in those situations. Well, thank you so much for showing up this evening. I want to point out that I have a few letters at the end of my name. This is not an MD, so this is a PhD, and which means that I will basically give you a biology seminar. And basically what I do for my freshman college class, this is what I will present for you this evening. Also, this also means I will not cover clinical data concerning COVID. I will just stick with within my expertise and do the biology of the virus. So let's start from the beginning. What is a virus? And we have a nice size comparison here. On the left side, this is a human cell, which measures around 100 micrometers. So micrometers is 10 to the -6. You really need a microscope to see them. In comparison, next, we have the next size, different types of bacteria. Bacteria are different sizes and shapes, but they do measure around 10 micrometers. And then lastly, down here in this corner, you notice the smallest of all. These are viruses. Viruses are between 0.05 and 0.2 micrometers. So, 10,000 times smaller than a human cell, around that range. In biology, we like to put things into categories. So everything that is living is classified into one of 3 different domains. We have the eukarya, the archaea, and bacteria. And these 3 are further classified into 6 various kingdoms, which incorporates all of living things that are on this planet. So we have here eubacteria, a type of bacteria, a different type, similar bacteria. We have protists, plant, fungi, animals. What you notice is that from this list, you do not see viruses. Viruses do not belong in the classification of living organisms. It's very important. So we do not consider viruses as living organisms. They are just particles. We refer to them as particles. And why is that the case? Because viruses do not have the machinery that other cells have or that organisms have. Here on the left side, we have a depiction of a human cell. And you'll notice that inside there are a whole bunch of compartments. And each of those compartments function as a type of factory. They each have a specialized function and they produce something. Here on the right, we have a virus, a viral particle, and you'll notice how simple it is. First of all, there are no compartments inside that are similar to the factories that are very prevalent in the human cell. Okay, you have some genomic information, and then some protein on the outside, and some fats. Whereas here in the human cell, here in our nucleus, we do have our genomic information, but a whole bunch of extra stuff. So human cells, they have genomic information, 2 different kinds. They have DNA and RNA, both present at the same time. Viruses are different. They can have either DNA or RNA, but not both at the same time. It's an either or. But they both have genomic information. So since viruses are not living, they require a host. They require something else in order to multiply. And I like to use the analogy of a battery. Batteries on their own, they do not do anything. However, as soon as you plug in a battery into a toy, it gives off power and that toy is functional. Viruses are something like that, similar. So what is a host? Viruses can live inside bacteria, animals, humans, anyone, either one. Plants as well. And a virus, it is selective. So depending on the type of virus it is, it will infect selectively. So a virus will infect certain types of cells. Now, in science, we have something that is called the virome and the microbiome. And this refers to the total collection of bacteria and viruses that are inside your body and on your body. And this is the normal, healthy individual. We're not referring to someone who is sick. And it turns out that bacterial cells, if we look at a ratio of bacteria cells to human cells, for every 1 human cell, you have 10 bacterial cells in your body. And that is normal and that is healthy. And you can guess where I'm going with this. How many viral particles are present in the human body? And you could probably guess a ratio in your mind before I show the answer. Viruses themselves outnumber bacteria 10 to 1. So basically what you have is 1 human cell and 100 viral particles. How is that possible? How can you have more bacteria or more viral particles than your human cells? It's the size comparison. If you go back and remember the size. They're so very, very small that you can fit a whole bunch of them inside your body. So you'll notice healthy people are loaded with viruses, and the estimates are at 380 trillion viruses living in you right now as a healthy individual. It really helps you understand what it means to be human, right? It's a different dimension. So we already are familiar that bacteria and viruses, they promote disease. That's what we're familiar with. However, they also promote health. So for example, we need bacteria inside our gut to help digest food. They'll also help in the production of certain vitamins. And viruses also, they have a good function as well. They will kill bad bacteria. So if we inhale or get infected with certain types of bacteria in our lungs, good viruses will infect the bacteria and kill it. So they're beneficial for us. So there were a group of scientists that did a very interesting experiment. What they did is they took a bunch of patients and they analyzed the blood plasma to see what kind of DNA is floating around in the human blood plasma. And what they noticed is that most of it belongs to viral genome. And if we take this figure as a graph, they found out that about 68% of the DNA that is floating around in the serum is from viral origin. About 10% belong to bacterial origin, very small amount, 3.5 to the human host. So a lot of the genomic information in your blood plasma is of viral origin. So we are interested in a specific class of viruses, the coronaviruses. And when we say coronaviruses, it refers to a family of viruses. We know that coronaviruses require a host. And the host in this case, the primary hosts are bats and rodents. That's where they reside. Now, from the bats and the rodents, these viruses have been passed on to a secondary reservoir. And we have so far identified these ones, okay? For example, the civet, raccoon dog, pangolins, dromedary camel. These are reservoirs for coronaviruses. Now, there are a number of different coronaviruses. They belong in the same family, and they have specific names. Okay? These ones that are highlighted in red, they have created previous epidemics. So, for example, in 2002, the SARS-CoV virus, there was an epidemic that was started in 2002 based on that one. In 2012, there was an outbreak of the Middle Eastern Respiratory Syndrome coronavirus family in the Middle East. And today we are familiar with this specific one, SARS-CoV-2, that was identified in 2019. Just a little history, the MERS, it had a mortality rate of 40%. Okay, the epidemic was in the Middle East, and the reason it was able to be contained is because when someone was sick or infected with that virus, it was visible. Those people were isolated and the animals were killed. So then that virus was able to be contained. Unlike today, when someone is infected with SARS-CoV-2, somebody may be sick, but they're not sure, and they'll walk among the population and spread the infection. So a bit different, different display. So like I said, these three caused pandemics. What is interesting to know is I highlight here 4 different types of coronaviruses. These ones are endemic as a cold virus. So when you have a common cold, you may have one of these coronaviruses. They are in the same family. What does it mean to be in the same family? For example, I am a Stanculescu. We have 5 children. Our children share our genetic information, but still they are unique individuals. And it's the same way with the coronaviruses. There is the parent and then the different types within that same family. So what happens when a virus infects a host? Some interesting things, actually. Viral genes get shuffled inside a host, and it's possible to produce a new viral strain. So for example, here is our host cell. We can say cattle or swine. And if you have 2 different viruses infect the same host at the same time, you can have a reshuffling of the genetic information. The genome recombines, and you have the production of a new viral variant. And what is the outcome of that new viral variant? Well, it's possible that that shuffling will cause that virus to transmit to humans. So originally, that virus may have been only endemic in the animal population, but because of the reshuffling, it is now possible to infect human species. Also, a new viral strain will evade the immune system. So the host will not recognize it and will not be able to mount an immune response against that viral strain because it is new. So let's take a closer look at the coronavirus that we are specifically interested. This is a 60,000 times magnification with a very powerful microscope. And this is a computer buildup of an X-ray. What we do is we crystallize the viral protein, and we're able to generate a computer image of it. You'll notice we have these blue hands sticking out from the edge. We call these spike proteins, the blue hands. And then there is a white envelope surrounding here, a sphere, that is made out of lipids, which is just a fancy word for fat, a whole bunch of fats. And then there are some red and yellow portions here. These are membrane proteins. They help and maintain that shape and that envelope structure. So what if we take that viral particle and we just cut it in half? What's inside it? Here's a cross section. Here are our spike proteins that are on the outside. We have the membrane proteins that are dotted throughout the outer lipid membrane that help maintain the shape. And then inside it's very simple. There is some genomic material, in this case RNA, and that genomic material is surrounded by a whole bunch of proteins. And that protein is called nucleocapsid. You'll notice the size of this particle. We are on the nanometer range, 10 to the negative 9. Very, very tiny. So genetic information can be DNA or RNA, but this particular virus carries only RNA. If you are using those at-home kits to detect if you are sick with coronavirus or not, the Abbott, Quidel, and Bectin, they are all testing for the presence of this particular protein, the N protein. So the protein that is inside that viral particle. Let's look a little bit at the naming system. So SARS, SARS-CoV-2, this is the name of the virus. And each of those letters stands for something. S, severe, A, acute, R, respiratory, S, syndrome. And then CO is corona, V is for the virus, and 2, this is the second coronavirus of that kind that we detected. So COVID-19 refers specifically to the disease. And that also is built up from CO is corona, VI, virus, D, disease. It was detected in the year 2019, so it gets that 19 designation, COVID-19. Okay, so we know what a coronavirus is, what it looks like. What does it do to our body? And here is a picture that shows that this virus can infect your human cell. And when it infects your human cell, it has 2 different routes by which it does that. We call the first route the direct plasma membrane route, and the second one the endosomal route. So the direct one, here is our viral particle. It has these little hands sticking out, and we like to say those hands are like a lock and a key. It has a whole bunch of keys on the outside, and it is looking for the specific lock on the human cell, on the host cell. So the lock comes in and the key comes in and looks for the lock, which is called ACE2. And when they bind together, it triggers a change in the shape. And there's another little guy here called TMPRSS2. He is like a scissor. He cuts something here and it allows the fusion of the viral particle with the human cell. And then you have the genomic information being spit into the human cell. So the viral particle has just infected that human cell. A different route that this viral particle can enter is called the endosomal route. And in this case also, you have that key, the spike protein, looking for the lock, ACE2. Once it binds together, it triggers the human cell to engulf the entire particle. So it kind of like eats up that entire particle, and it comes inside the human cell. And then once inside here, notice it's still inside a compartment. That compartment needs to be opened up, and that genomic information kicked out into the human cell inside. So this is one reason why some medications don't work. You know, if somebody is specifically taking, let's say, chloroquine will block this route, but it will not block this route. Okay? Now I say here, under normal conditions, this ACE2 lock is expressed in capillary-rich organs. What are those? Some of those examples: the lungs, kidneys, the gut, the brain. They normally express that particular lock. And here is just that naming system. So the spike protein is like a key, ACE2 is like a lock. What happens under certain disease states? You know, if someone is diabetic or they have cardiovascular disease, unfortunately what we see is that there is an over or increased expression of ACE2 in the heart. So in your heart cells, you will have many more of these locks that are being produced in your own cells. So if you have a lot more locks, that means there's going to be a lot more viral particles that are going to bind there. And that is an unfortunate reality, why people with comorbidities have a harder time getting through a COVID-19 infection. Okay, this is a busy slide, but I really want to walk you through it so that you understand, what does that virus do when it enters your human cell? Why are we so upset with that specific event? So everything that is, here is a double line here. Everything that is under the double line is inside your cell, your human cell. Here on the outside is where the virus particle is coming in. So here we have our SARS virus. It's coming in. It has a ton of keys, and it's looking for that specific lock. And it's going to scan your body. It's not going to enter any cell, only the cells that have that expressed. Once it binds, the virus particle is going to enter your human cell. That membrane, the viral membrane, is going to open up, and it's going to release its genomic information into the human cell. Once this enters in, this is where all of the human factories kick in. So the human cell has all of those small little compartments and factories. They know exactly what to do with genomic information. They feel it, they sense it, and they immediately come and surround it and start to produce new viral particles. So all of these busy steps, 4, 5, 6, 7, the cell is just starting to pump out a whole bunch of new viral particles. It will start copying the original genome from the virus and make many of its copies. It will produce proteins so that it could package that genome into a viral particle. And then you have production of a whole bunch of new viral particles. Now the cell, it will keep producing them until it dies, because at some point it can't resist anymore. It's gonna kaput. So it'll keep on pumping out a whole bunch until this cell itself is depleted. So we call this the viral life cycle. Okay, this is the life cycle of a virus. Enters the human cell or the host cell, replicates itself, produces a bunch of new particles, and it's released into the outside space. What happens when it's released to the outside space? It goes and infects other cells. So let's take a look at what happens if a viral particle infects your lungs. If we take a look at our lungs, we have 2 different lobes. And each of those lobes, they are actually made up of a whole bunch of branches, like on a tree. And those branches, they branch out until they get down to very fine, thin branches that end up in what looks like clusters of grapes. These are circular sacs, and there are millions of them inside your lungs, in each of your lungs. These sacs, the whole purpose is to absorb oxygen and transmit it further on into your body. So here is some tissue taken from a patient that is comparing normal lung tissue to tissue from a patient that was infected with a SARS-CoV-2 virus and has a high level of lung disease. Notice the normal state. So these are those circular sacs that we saw on the previous page. Notice that we have a very thin boundary, okay, all around, and a whole bunch of white space. That white space is where oxygen is present. Oxygen enters, and then it's absorbed by these cells and transmitted further on. Now, in this unfortunate patient on the left, you'll notice that you no longer have narrow boundaries. They have been grossly thickened many times over. So what happened is that the virus entered, infected these cells, killed them, and when you kill cell, the immune system is stimulated to come and try to repair the damage. So you have a whole bunch of cell debris, cell garbage, and the infusement of immune cells to this local place. And so you have gradual thickening of that region. So you can imagine that this person is not breathing well. There is no space for that oxygen to fill up with oxygen. We don't have those empty spaces anymore. And also, oxygen no longer diffuses across readily. So the process has been slowed down very much. So this is what happens to your lungs when that virus infects your lung tissue. I do want to point out down here, notice it says same magnification. Okay, the left and the right is the same exact magnification. So you could really appreciate the destruction of the tissue. So then in that case, we learn about the virus, what it does, how it infects your cell. What are variants? What is a viral variant? So variants, they are actually just small changes in the shape of the virus. So again, here is our virus of interest. It has those red spikes. The color is, generated by the computer. It was green on the previous slide, here are the red. So these spikes, if we take a look at them and zoom in, they are made out of amino acids, so a whole bunch of small little building blocks. And every small little circle is probably too hard to see, but they each refer to a different type of amino acid, so a different type of Lego piece. And you build up your spike protein. Notice that some of these Lego pieces have a specific color. We have blue, we have some red, a whole bunch of red up here. Those red dots are telling us these Lego pieces have changed when we compare the Delta virus with the original virus that came out of China in 2019. So this is the Delta version of the virus. It has these specific changes. Now you notice that we have the Omicron version on the right. And immediately you'll realize that there are a whole bunch of extra red dots. Okay? And what that tells us is that the virus has changed in those locations over time. More importantly, you see that those changes kind of aggregate in a special location. Notice this top here that is highlighted in yellow, and here over here. You have a whole bunch of changes in this yellow portion. That's important because that is the receptor binding motif. So if you think about your key, the top of the key is that yellow spot. And the top of the key, when it enters your lock, if you're changing that shape, you're not going to be able to unlock your house door any longer, much longer, will you? The key will, it'll be okay for maybe a small little rub of some sort. But eventually, changing the shape will affect the way that key is going to open the house door. So this is what we have here. Omicron has many more mutations or changes in its shape compared to that original Wuhan strain that came out in 2019. So just to repeat again, Omicron has more than 50 mutations compared to the original Wuhan strain. What do these changes do? What happened to our virus? Well, it caused Omicron to bind more tightly to ACE2. So that lock was getting much more tightly binding there inside that lock. It also caused the virus to evade the immune system. So our body was no longer recognizing it. We may have had the original, we were probably sick with the original virus early on, but that receptor part changed so much that our body is not recognizing it. So our body is responding with an immune response. And you may get sick again. So when I looked at some of this data online, back in March 2022, over 2 million Omicron genome sequences have been uploaded in a worldwide database. So there is a database called GISAID, and people from all over the world upload sequences up there. These sequences are coming from patient samples that are collected by the hospital or sent to a laboratory. The genomic sequence is figured out, and you can go yourself to that database and search it and see the type of information that is uploaded there. Why do we collect that type of information? So this is a graph that I pulled off from the CDC website. And what we notice here is that we have these bar lines with different colors. Here on the y-axis, there is a percentage, and it says percent viral lineage. Across the X, we have each bar refers to a week of the month. And it starts out with December 2021. So back in December 2021, almost 99% of all of the sequences that were being collected belonged to the Delta variant of the virus. So December 2021, 99% of the virus circulating was a Delta variant. Already by March 2022, which is this bar right here, notice the changes in color. We no longer have that yellow version. Rather, we have this maroon color with some purple and light purple. They refer to the different variants that are popping up in the sequence genomes that are being sent from the hospital up into these labs here. So by March 2022, 100% of the circulating virus belonged to 3 different kinds of the Omicron version. And here is a chart that further classifies it. In science, we have some more complicated names to try to keep track of the different variants. You'll notice here, Omicron can be this naming system. There's a B.1.1.529. So we have a different type of naming system to be able to keep track of what's going on here. So, okay, we're collecting these sequences. We know what kind of variants are out there. What else do we do with that type of information? And this is a screenshot from the CDC portal, and they try to annotate the different variants with specific information that is of interest. For example, this particular one is for iota. So iota is the label that World Health Organization gave it so that the public can follow along. The naming system that we will use in research is B.1.526, so it gives us a very specific naming system. And this particular variant has been already annotated. Notice that we have some information here. It tells us the substitutions or the changes that have occurred in the spike protein. So here it's telling us the iota coronavirus variant has these changes in that key, surface of the key. And did that, okay. And they tell us that that particular one was first identified in the United States, in New York, in November 2020. Okay. And we have some clinical data associated with it. For example, we know that this particular variant has reduced susceptibility to the combination of a certain treatment plan. So we're trying to get a more comprehensive understanding of the variants, their response to medication, and associate that with the different changes in that spike receptor. Actually, hospitals are required to submit a certain number of coronavirus sequences from the patient on a weekly basis. So this is a huge database of information that is being collected. Okay, so what can we do to protect ourselves from this virus? First of all, only antibodies effectively neutralize the virus. So here in this picture, here is our virus of interest. And all of these Y-shaped figures are antibodies. Antibodies are something that our immune system will produce in response to a pathogen. You get sick with bacteria or virus, your immune system will start pumping these out. These will bind specifically to the virus, and notice what happens. If these keys are blocked off by the antibodies, it will no longer be able to interact with that lock. And if you block off that interaction, the virus can no longer enter the human cell. So you have basically stopped infection at that point. Where do we get antibodies from? How many sources do we have? It turns out that we have a few sources for antibodies. For example, vaccination is one way to be able to get antibodies inside your body. And this is a prevention. So your body has not been exposed yet to the virus. Vaccination will stimulate your body to produce these antibodies. A different way to get these is through natural infection. So if you were naturally exposed to the virus, you got sick and you got better, your body will naturally have antibodies and produce antibodies to that specific virus. We do have some commercial products. For example, pharmaceutical companies will produce commercial antibodies specific for the virus. And you can also get them from convalescent plasma. So people who were sick with the virus, they recuperated and they want to donate blood. That's another good source for antibodies. There is a different way to try to block that connection between the virus and the human host, and that is through small molecule inhibitors. So these are basically small chemicals. They try to interact with either this receptor or this receptor and try to block off that interaction. So we do have medications that target various parts of a viral life cycle. So here is just a visualization of some medications and how they interact with the human cell or the virus. There are many, many more. I just have a few here to kind of give you an idea of what's going on here. So again, here we have a red boundary. Everything on this side of the boundary is inside your human cell. And here is outside the human cell. Here is our viral particle of interest, SARS-CoV-2. Notice that it has to bind to the ACE2 protein in order to get entrance of that viral particle. Notice we do have some types of medications, Arbidol or Umefinovir. These will interact and try to block off that interaction between the receptor and the lock. A different type of medication, Camostat, this will interfere with this tempers protein, which acts as a scissor, and try to block off the function of the scissor so that it doesn't activate that connection there. Down here we have Ivermectin. It has also been shown to interact with that molecular scissor and try to block it off so that that interaction does not take place. Once you have entrance of that viral particle, it is surrounded by a membrane, and that membrane needs to open up and release the genetic information. However, it has been shown that chloroquine will kind of block that opening of the membrane and kind of just get that virus particle stuck inside that little compartment. Further on, here we have the factories of the human cell that are trying to produce extra viral particles by copying the genome and by producing extra proteins so that you could get a new particle. We have various medications here, for example, lopinavir, oseltamivir. We have remdesivir. Remdesivir is very popular in the clinic. It will interfere with the factory so that you don't have that production of more genomic information. Also, you'll notice that ivermectin also interferes with the replication of the viral genome. Chloroquine also seems to interfere with the proteins that are involved in replicating that genome so that you don't get more of that copying mechanism in place. There are many more medications on the market, but that's just an idea of how these chemical inhibitors work. So all of these medications, they have activity that is non-specific to our virus of interest, and this is why we have side effects occur. So for example, ACE2, this lock right here, it was present on normal cells way before the virus ever showed up. So it had a certain function. And ACE2 actually is involved in regulating blood pressure. So if you have an inhibitor of that receptor, you will greatly decrease your blood pressure, which may be fine for people with high blood pressure, but in other cases it may not be fine. And it could actually be dangerous by slowing down the heart rate. So medications have side effects because they interact with more things than just one. Okay, can vitamins block the virus? We all know that taking vitamins is good for you, but how exactly do they work? The way it works is that vitamins strengthen the immune system, which in turn clears out the virus. When we talk about the immune system, we're actually referring to a whole bunch of different cells. And here we have a picture. The immune system is made up of many different cells, not just one. So for example, vitamin D. First of all, vitamin is a misnomer because vitamin D is actually a hormone. However, It's very good for you, and it has been associated and shown that it strengthens the immune system. So vitamin D has a receptor, again, that lock-in key combination. In this case, vitamin D is the key and the lock is the receptor. And the vitamin D receptor is on many cells of the immune system. So what happens is that vitamin D will go bind to the lock and activate that cell. And that's exactly what you want when you're sick. You want those cells to be active. And it has been shown that high levels of vitamin D are associated with tight control of the inflammatory response. So you really do not want overactive inflammation, overactive immune system. You don't want that. It's going to destroy your tissue. You don't want a silent or underactive immune system. You want it just right. And vitamin D is active in helping to regulate that response. How about vitamin C? We also know it's very good for you in many different ways. The way vitamin C works, similarly through that lock-in key combination, it has been shown that it can temper excessive immune responses, which damage organs. So again, you don't want a highly inflammatory situation because inflammation will destroy your healthy tissue. And vitamin C will help temper that. How about zinc? That has also been a very popular one during this pandemic. Zinc has been shown to regulate the ability of immune cells to clear out the pathogen. So there are different types of immune cells. Some of them act like a Pac-Man type. They go and they eat whatever is foreign, and zinc seems to be active in stimulating those type of cells. Also, zinc is required for the activity of many enzymes. Enzymes, they do work in your body, and some of those enzymes are necessary for viral replication, allowing it or blocking it, and zinc seems to be doing the good parts of many of those processes. Okay, so now that we talked about medication, about vitamins, how about the vaccines? Just want to explain what are they and what do they look like. So currently here in the United States, there are 3 different kinds that are available. There is the J&J, the Pfizer, and the Moderna. The J&J is the traditional type, the one that we have been familiar with for decades. It is based on a viral vector. So basically you have a weakened virus that carries some information, and you get vaccinated, and that information stimulates your body to produce an immune response, to produce antibodies. The J&J, it has an advantage because it induces a strong response. Your body will react and start producing antibodies. However, it's produced in an aborted cell line, and it caused some controversy among people whether they want to use it or not. 2 different kinds are the Pfizer and the Moderna. These are completely new vaccines that have not been used before. They're just new with this pandemic. The advantages of them is that they're very fast to produce. They do not have a viral portion, so they do not have an infectious particle. So very different from the traditional one. Also, these 2, they are produced by bacteria in robotics. They do not require a cell line the way the other one requires a cell line. What is an aborted fetal cell line, since the J&J has been produced in one of these types of cell lines? So sometime in 1985, some person with, what he did is he took an aborted fetus, I don't know from where because it was illegal, obviously, back then, that was about 18 weeks old, And what he did is he went to the back of the eye and scraped the retinal cells. These are cells that are behind in the back of your eye. He took those cells and he grew them in a plate. And doing that creates a primary cell line. When you take live tissue from any person, they will die after a few weeks. They're not made to last long. So because of that, what they did is they transformed it and made a cancer cell line out of those cells. And cancer cell lines are immortal. They basically live forever. And they're still around. Notice, it was started in 1985 and we still have them today in 2022. So it just goes on. So I just want to clarify that we don't have hands and feet that are being cultured or like body parts. The original intention was evil, but I just want to show you that they are cells that have been changed into a cancer cell line to go on forever. So how about a visual comparison of the 3 different types of vaccines and an ingredient list? What are they made up and what are they? Well, first of all, the J&J is the traditional one. So basically you have a piece of genomic information that tells your body to produce the spike protein. And when your body produces it, your immune system will react and produce antibodies. So here is our viral particle and that piece of genomic information. The J&J uses incompetent virus, which means that virus cannot multiply itself and reproduce. It's just there to carry the information that was necessary. So it contains the DNA instructions for the spike protein. That was our key, those hands that were waving on the virus. That vaccine also contains sugars and salts and acid. All of these are necessary for the stability of what's inside there and to keep that viral particle stable. They also have an extra ingredient, polysorbate, which is a potential allergen for some people. Polysorbate is in your food. So if you like to eat ice cream, it is an emulsifier, making that ice cream really smooth. So how about the Pfizer and the Moderna? This is what it looks like. Basically, you have a layer of fat on the outside. And that fat, it enables this particle to enter into the human cell because the human cell is made out of a layer of fat. And you know, if you have greasy hands, you want to wash your hands with detergent, fat dissolves fat, and they come together. These vaccines also, they contain sugars and salts for the same reason, to help keep stability of that structure. And it has mRNA as the genetic component inside here. And that is the information, again, instructions for the body to produce the spike protein. An extra ingredient they do have is PEG, which is a potential allergen in some people, in some portions of the population. PEG is an ingredient in laxatives, so it's not a chemical that we're not familiar with, probably. So here is a visual comparison. Here is the J&J, here is our mRNA. This is the SARS virus. And what I want to pull up here is something called an extracellular vesicle. So our normal cells inside our body, they make these vesicles so that they can communicate with each other. If we have a cell on one part of our brain and it wants to communicate with the other part, it will send hormones, but it will also send vesicles across to carry information. These vesicles are made out of a layer of fat and they have pieces of genomic information inside it. So it looks very similar to our mRNA vaccine. They're both made out of a layer of fat and some parts of genomic information inside. So in a way, the mRNA vaccine was modeled after a physiological mechanism. So extracellular vesicles, these are a normal mechanism to transport cellular components. Now, here's another question that I've heard a lot of people asking and discussing. Do mRNA vaccines change your DNA? So in humans and animals and all other organisms, there is a normal flow of information. Information starts out as DNA, and DNA is then copied into RNA. And then RNA is copied into protein. That is the normal flow. There are very few instances where we can see a reverse. Very few instances where RNA can be copied into DNA. Okay? Protein being copied into RNA, we've never seen that. We've been studying it for 100 years. That has never been seen yet. As for this into that, very few situations. It does occur in retroviruses. Retroviruses are a special type of viruses from which HIV belongs in that category. These viruses, they have a special protein that is able to copy RNA into DNA. A second situation is something called a human protein line. So normal, healthy individuals, that protein is not being expressed. It is silent. However, it is activated in cancer cells. What does that protein do? It can reverse copy any RNA that is present and copy it into DNA. DNA is the original genomic information that our body uses to store information on a long-term basis. DNA here, it lasts like forever, for the lifetime of a cell. Some cells live for years, many years. RNA is not stable. Maybe 2 minutes. The average mRNA molecule lasts 2 minutes. It's not stable. So our body always stores long-term information in DNA. So what I'm showing here is that there is a protein that is activated in cancer cells that can copy RNA into DNA and possibly get that piece of DNA inserted into the normal human genome. This is one reason why some people, they test positive for the virus even though they've cleared it out a long time ago. Okay? The laboratory test is still picking up a certain sequence. Now, what I want to clarify here is that the reverse copy is not specific to vaccine RNA, rather any RNA that's present inside the human cell. And we have tons of it all the time. Also, any of the reverse copy that has occurred, we have only seen evidence of small little pieces, not enough to regenerate an entire viral particle. Lastly, usually cells that have been infected, meaning reverse copy of DNA that has been implanted into the chromosome somewhere, these type of cells usually die because the cell has a whole bunch of sensors to detect something is not normal, it doesn't look normal like what we're used to. And these cells, they kill themselves usually. Okay. So all of these types of information, research articles, they're all published and kept together in a database that we go to called pubmed.gov. So this is not owned by the government, but the government provides the funds to maintain this database. And you can go to that website and put in a keyword of interest, and it will pull up a whole bunch of articles that you are interested in. So what I did back in March 10, I put in a keyword search, SARS-CoV-2 or COVID-19. And the writing is very small, but 190,470 results popped up. These results, I want to clarify, are research papers. So this is 190,000 research papers that were compiled from laboratories from all over the world, from Australia, from United Kingdom, from Canada, from China. Anybody who was engaged in research, they publish a paper, send it to a journal. There, it is read by a number of people. They want to make sure that you did the experiment right and that you pulled the correct conclusions, at least based on your experiment, and then they will upload it into this database. Most of the articles are free and they're available for you to read. So guess what I did about 2 days ago? I did the same keyword search in the same database, and can anybody guess how many articles popped up? 1.9. Wow, that's pretty impressive. Not quite there, but very impressive. So we went from 190,000 to 252,613 results. That's how fast information is being generated and deposited into this database. It's an extreme amount of information. I certainly have not read 200,000 articles. Which takes me to my next point. Okay, maybe many of you are familiar with this cartoon. What we have here is an organism, a very large one. And we have a whole bunch of men. And these men are blind. And they are each feeling around the organism where they are situated. This particular guy is feeling the tusks. He's like, wow, I think I am touching something shiny and smooth. It may be a piece of furniture. This guy says, no way, I am touching something hairy and rough. It must be an animal of some sort. And then each person is trying to identify what he is actually investigating. And they each have their different perspective. So this just reminds us that we really need to be humble in talking to each other and approaching this whole COVID pandemic with a spirit of humility. No one is able to have 100% of the right answers. We try to come together and have respectful conversations, trying to just understand more and more of what exactly is going on. I do want to point out, how does this relate to creation? Well, viruses evolve and they are still viruses. So people use this word evolve very loosely. Okay. Here I just have a picture of the alpha coronavirus, the beta, the gamma, the delta. In comparison, they are still coronavirus. Okay. It's the same thing. Mutations, the small little changes, they do not produce new information. So for example, here on the right, I have a picture of the spike protein, that key, of Omicron, which has 50 new mutation sites. It evades the immune system just because the shape here has changed so much that the antibodies don't recognize it anymore. Okay. So what that caused is that Omicron was more contagious, and in most cases the symptoms were less serious. However, it is still SARS-CoV-2. It will never become a bacterium. It will never be able to evolve into even a bacterium, which is pretty tiny and 100 times larger. I do want to expect that we can expect more zoonotic diseases in the future. So zoonotic diseases means diseases that were that are endemic in the animal population, but they hop over into the human population. And why? Because the more we encroach on animal habitats, the higher the prevalence is for endemics or pandemics and so forth. I would like to close trying to explore the idea of why would a good God allow suffering? So we know from the beginning that God created the world perfectly. And in Genesis 1, it tells us, God saw all that he had made and it was very good. God was pleased with what he had created. However, because of Adam's sin, God cursed the earth. Genesis 3, cursed is the ground because of you, Adam. That was not God's intention. Praise the Lord, Jesus Christ had a plan. He came to restore what was broken. And in Revelation, we have this beautiful verse. He who was seated on the throne said, I am making everything new. Then he said, write this down, for these words are trustworthy and they are true. However, Christ is king, but his kingdom is still contested until the culmination of his reign. So Jesus' reign has not culminated. We are still waiting for that final day when he will. And here in Revelation 11, we read, the kingdom of the world has become the kingdom of our Lord and of his Christ, and he will reign forever and ever, uncontested. And we do wait for that one day. Thank you so much for your attention.